The first patient is from,
Oh, here, I can have that part
it's
of
a,
it if it
okay,
works
it's
for
a,
you.
it's a BAL, and it had a partially acid-fast thin branch filament in it, and we ran it through the Nocardia database. And we do, with the Nocardia, we have two sequencing primers. We have one we call NOC, which is the one that works on most of the organisms. And then there's two organisms that it's sort of, the NOC wants to group them together. And one of them is it Nocardia farcinica?
Mhm.
Is that how you say it? Nocardia farcinica. And it is the one that can be resistant to Bactrim, which is used as a prophylactic agent and to treat a lot. So it's sort of important that you tease this organism out. And the organism it is lumped with is Nocardia series C. georgica. So then they developed a second primer, which is the NOC1, which its total job in life is to differentiate
Those two.
Nocardia farcinica from Nocardia series C. georgica.
There you go.
And so you never look at NOC1 if the NOC doesn't tell you it's one of those two organisms. You just don't look at it. So this one, the NOC told us it was Nocardia otitis caviarum complex, or
These are really
Nocardia,
mouthfuls.
or Nocardia nova complex, or Nocardia transvalensis complex.
You
And
need
it's-
to see the names, you can see them here written out, just to see what they look like.
And those organisms, well, at least two of them can be separated if you just read out 36 base pairs instead of 20. So we read out 36 base pairs, and we're going to sign it out as Nocardia otitis caviarum complex or Nocardia transvalensis complex.
Okay.
No, I'm sorry.
No.
Pay no
I think-
attention to me.
yeah,
We're going to
that's
sign it
right.
out as Nocardia
Nova. Transvalensis.
or transvalensis.
Yeah. Right.
It's not
Right.
not Nocardia otitis
This
caviarum.
is Dr. Miranda's patient. So, this is a VAL with
Yeah,
what?
lung
With Nocardia.
transplantation.
This is Matusik, so yeah, that would be right. Yeah.
It's
Yeah.
likely.
Right. And then, has she asked for a susceptibility on this already?
Yes, she has.
Yeah,
So
okay.
that's in mycology. I think that's going out to Dr.
Wallace's lab. So we send out all of our susceptibilities to Dr. Wallace's lab in Tyler, Texas. So that's probably going to be on its way out. Interesting. Good. So, and we see Nocardia throughout all of the year, but there's no doubt Nocardia is one of those organisms that you tend to associate a little bit more with warmer climates, so maybe a summer bug. Now this year is ridiculous because it's summer all the time, so, we have a lot of Nocardia, I say. No doubt about it. Hello,
I'm
Maria.
sorry.
That's all right, that's all right. So we'll go through some of this afterwards with you individually, but this is the first one that we did today, and it is a Nocardia nova, Nocardia transvalensis. And our method won't get farther than that. Most sequencing methods probably won't get too much farther than that either. Although we sometimes, when we send it out for susceptibility testing, the group down in Texas will give us a hint of what they think it is. Or you can pay them and they can tell you specifically what they think it is. So I think this is probably gonna be good enough for Cindy. I think it'll be good enough for Cindy.
If it's already gone out for susceptibility testing, we can just call them up.
Call them up if she wants a
If she
further
wants it.
differentiation on that, if they can do it, yeah. So that was 100%, this one worked.
Yes.
A BAL from Dr. Budev, so another, I'm sure, screening one. And this was positive AFB, looks like a rapid grower.
Right. We were expecting a rapid grower. It grew out on the CYE plate of a Legionella culture.
Okay.
So we were expecting a rapid grower, and it is Mycobacterium chelonia abscessus.
All right, so we do have a rapid grower. Now, are you familiar with, probably not every single mycobacterial species, but we've got TB, we've got MAI, we've got Kansassi, we've got Marinum. Those are sort of the big four. And then we've got tons and tons of rapid growers that we see, and Chelonia abscessus is probably the most. Next one is a sputum on a patient from the clinic, G62, and it looks like it's had some problems.
They thought they saw two different colony types, a large and a small. So I sent both of them through. It came up Mycobacterium mucogenicum complex, but it was only 93.7%.
You would think that would be good enough, right? It's not
And
for
it's
our
not.
assay, we don't accept it.
So a lot of times when they're like this, they're probably our rapid grower. So I blasted it in.
Oh, now she's going to show you the blast, all right.
Yeah. It was quite nice, except you don't get to see it in color. And it was one of those Mycobacterium concepcionensis and
Mycobacterium
fortuitum,
Mhm.
and I think I've gone through here till 60 was done. I'm not even gonna try some of these names.
Okay, Senegolens, yeah.
And,
Senegal,
um,
yeah. And all of these have what I call 60 bits, so when she blasted into GenBank, the score, or similar to a score, if it's 60 bits, then you accept it. It's much like 100 here. You can accept it, but if it's not, then you don't accept it. But then when we get 3 answers like that, which one of them is it? Is it any of them, kind of thing? So what are we left with now?
well, in the past, what we've done is, we've repeated it, and if it repeats, and since we did it twice
Mhm.
here,
Right.
I'm sort of thinking we might be done.
Right.
We have a code that says rapidly growing mycobacteria, not Chelonia abscessus.
Right, and that's probably as far as we'll go on this one. It's a sputum, hard to know. It could be relevant, certainly, and if we've got it in a lot of cultures, but I don't know that we do or not on this one in particular. And we basically wait for the clinician to tell us, and if a clinician should call and say, absolutely, you have to know what it is, we'll send it out to some other lab that might be able to, or long sequencing or something. But in this case, most of the time when Mary's done this, people accept it. It's a rapid growth. It's not TB, it's not MAI, it's not Kansas-City, and that is usually sufficient for them. But we're not going to get any farther by repeating this on and on and on.
So usually that blast is done whenever you are encountering atypical or a mycobacteria or maybe a nocardia that's not part of the database.
Exactly.
Yeah.
And
Yeah.
then you usually repeat it 2 times, maybe the week
We've done as
after
many
that.
as 4 and 5, but,
Yeah, well, we keep thinking we can get it.
yeah,
but
right.
This one, the sequences for the large and the small were exactly the same, exactly the same.
So it's not mixed, it is the same organism.
And
What's
what,
the quality like?
The quality is good,
Mhm.
1 through 44 pass, and since we only read 30,
That's
we're good
really
there.
good.
And then 45 through 66 for both of them were check, and 0 failed.
So there probably is a name in this if you were to send it for full sequencing. This is one of those organisms that 30 just doesn't make it. We can get this close, and we know it's a rapid, that's about as close as we can get. So whether or not MALDI will make the difference or not, so we talk about MALDI and mass spec all the time, whether that'll make a difference, that's the other job that Dr. Harrington's going to be working on with Mary and the group, is taking all of the mycobacteria that are well characterized, putting them through the MALDI and see how they come out, and then taking some of these others. But we'll run into the same difficulty that just because it says it is, you have to prove that it is, so then we'll have to take those and do full sequencing on those, and then we can add it into the MALDI database eventually, so that's yet to come.
What does blasting?
Oh, yeah, they in the gene bank database.
Oh, okay.
It's online. There's
Yes.
Yes.
that online for Gene Bank.
Mhm.
And Gene Bank has all these, it's a database of all the sequences for all these different types of mycobacteria species.
As well as all
and
kinds of
Nocardia
bacteria.
species and
The
all
human
other
genome?
kinds of
Yeah,
bacteria.
yeah.
So they have a blank that you put in your
You put
30
the
base
gene-
pair
the nucleotide,
sequence,
yeah.
and then it will go through its database and find
A match.
a match.
So you would enter
I
what
I-
you
I-
got
What you got.
from
Mhm.
the pyrosequencing.
Either what the machine told you, or if you think that your manual read is better, what your manual read is, and see where it says 60, those are the ones that will accept it. Usually you'll get 60s, it'll be one name, and it's like, yeah, that's probably what it is. But if you look through these sheets, there's a lot of 60s and different names for those 60 matches. So
Yeah, like
who
here.
knows which of,
Yeah.
right, which of those organisms it is, absolutely. There are so many rapid growers out there, it's phenomenal if you look at some of the newest taxonomic sheets. There are hundreds of these rapid growers now that are coming out, unfortunately. And there's data, like we said about, if you, or I said it this morning maybe, every organism, even ones that we always said was always a contaminant, you can find some article that was written someplace that said it caused the
There will be a
disease.
clinical
Whether it did or not, but you'll
correlation.
find somebody who wrote a report on it, so you can never say that either. But at this point, I think just saying it's a rapid grower will probably work. Now, sometimes we will go ahead and do a susceptibility, even though we don't know what the name of it is. It's a little tricky because some of the things you read based on what they are. But if someone really would want it, we could probably do a susceptibility, and at least it would help them to know if clary would work. Clarithromycin is usually the drug of choice. Okay. Next one is an outside. So we got this isolate from Akron General. It's a bronchial wash on a patient, and, oh, interesting one, okay, I'll let Mary take it now.
Okay, last week we ran it through the Pyrosequencer, and since we only do 30 base pairs, we do have some groups. And one of the main groups we have, or the one that gives us problems, is the 30 base pairs cannot differentiate between Mycobacterium kansasi, Mycobacterium scrofulaceum complex, and Mycobacterium gastri. There's also Simi, Conspicuum, Paraphinicum, and something called Habana.
So it can't differentiate amongst that whole complex.
So so what we usually do, if it's a respiratory site, the next Friday we do gen probes. And so we ran the Kansasi probe. 95% of the time, it is Kansasi.
This one wasn't.
This one wasn't. So what they did was when Dr. Prokop and Marion were developing the database, they saw this little group of organisms, and what they had chosen for their 30 base pairs was called the hypervariable region of the 16S. They just went down the 16S and found an area where you could differentiate the two, or actually the three, and developed another primer for that. So
That's
this
what
week,
you ran this
I
week.
just ordered up the sequence T
Good,
primer,
good.
and it came out to be Mycobacterium scrofulaceum
Scrofulaceum.
complex.
Which is a little bit unusual of a respiratory organism, unknown what its relevance is. It's the organism that we used to associate with lymphadenitis in little kitties. Now little kitties get more MAI and MTB than they get scrofulaceum, but still that relationship is there. So in the respiratory tree, there is no doubt Mycobacterium kansasi is a pathogen. It's hard to know whether scrofulaceum is a pathogen in the respiratory tract or not. I'm going to bet this patient will be treated, but probably, but it is an Akron General patient, if you will. So we don't know a lot about the history on that patient or anything at all. Although one of our techs who used to be working with Mary in her lab actually now works down there, so we could call and ask, I suppose. But anyway, good, scrofulaceum. We don't see very many of those. This one is an outside patient, another Akron. Oh, and then you worry about it when you get two of them in a row from the same place, and they're both unusual. So this one is bronchial washing as well. Let me look at the dates on these two. This one was collected on 6/27 and this one on 7/3, so not together at all. And this one, I guess, failed. Is that
Failed,
right?
no hits.
No hits. Interesting.
So we'll do, I did probes last week.
Okay, yeah.
And MAI was like a really weak positive.
Yeah, 41,000.
So I'll try it again. The only thing I can imagine, the reason for the no hits would be if I got a little bit of contamination in
Okay.
there. So I'm going to examine the plates for contamination too. Or
So
mixed.
you want to show them that one on there if you can?